Before we talk about methods for studying proteins, I want to make sure you can picture what the proteins look like in their natural environment. And I really love these illustrations by David Goodsell. On the left, you see a picture of E. coli cell with outer coat. You can see kind of the tree-like structures are carbohydrate coats, sugar coat, there's a flagella, and a variety of proteins below the membrane in purple and yellow. On the right-hand side, you see a fragment of red blood cell. You can probably recognize hemoglobin molecules in red. Also tree-like structures at the membrane. These are transmembrane proteins with sugar coats on them. And then in yellow, you see blood plasma with a variety of proteins, and this kind of, the blobs here are lipid particles that when we want to study protein structure or function, what we have to do is separate our protein, the protein we're interested in, from the rest of the proteins, right? And also notice it's really crowded here. Whether it's blood plasma or cell cytosol, there's lots of protein around, right? So, and they're different proteins. And if you look closely, and this is what's great about these illustrations, you can probably already recognize by shape some of the proteins. So the Y-shaped proteins that are showing up quite a bit, right? These are, hopefully you recognize them as IgG, antibody class G. Some of the proteins, like this five subunit structures, IgM antibody. The stringy things here involved in are structural proteins that are involved in blood clotting. And then we haven't seen this yet, the little V-shaped protein right here, that's actually serum albumin. And I put structure here of bovine serum albumin because it's probably the most commonly used protein in labs because it is easy to obtain in large quantities and cheap. Its job is to carry fatty acids through the bloodstream. So imagine fatty acids are nonpolar, right? And they would not be easily soluble in aqueous environment, but they end up sequestered inside the protein, up to 7 fatty acids per serum albumin molecule. So the methods we're going to consider can be divided into analytical methods that are used to kind of learn about the molecules but often destroy the molecules themselves, and purification method where we mainly focus on obtaining our protein of interest and as large quantity as we can, as pure as we can. When we do this sort of techniques, we can exploit different properties of the molecule to allow for that separation. So by charge of pi, you can do isoelectric focusing or ion exchange chromatography. By size, you can separate on SDS-PAGE gel, or for purification, gel filtration or size exclusion chromatography. And then affinity or binding to something is also a property we won't spend too much time on, but it makes sense with antibodies that can be really powerful to, if you have the antibody, to be able to recognize your protein of interest, whether it's in a gel or in solution. You can even do immunostaining right in cells if you're doing microscopy, or if you can put the antibody onto a column, you can do, or on some kind of resin, solid support, purify by affinity chromatography. I will say that, affinity methods are not only for antibodies. If your protein binds something you know about, then you can use that as well. So for example, DNA binding protein can be purified using a column that has DNA mobilized on it. So only DNA binding proteins will bind to it, and other proteins will kind of flow right through. All right, so let's review isoelectric focusing. It involves 2 components. We need 2 electrodes just like any electrophoresis, but in addition, we also use pH gradient. So when proteins are at low pH, they tend to get protonated, more positive, and move towards negative electrode. When proteins are near negative electrodes, they're also at high pH, so they get deprotonated and more negative, and as they become negative, they move towards the positive electrode, right? And eventually, they're going to find a spot where the net average charge on the protein is 0, even though there are some charged groups, right? And that's going to be the pH equal to pi of the protein. So at the end, you'll see a banding pattern, and where the band is tells you what the pi of the protein is. While on SDS-PAGE, we still use the mobility of charged molecules in electric field, but the separation happens by size, right? So make sure you're clear that in SDS-PAGE we separate by size, but the molecules still move due to charge. And when we do separation by size, our mixture of proteins has to travel through some sort of cross-linked matrix of the gel, and this the polymer of the gel kind of makes it difficult for large proteins to travel, while little proteins can travel pretty quickly, right? So notice this picture is a little misleading. The proteins should not be globular. They should be all unfolded and kind of linear. And the proteins are going to move and be charged because they're coated with SDS. Here's the structure of the detergent, sodium diadesyl sulfate, and it has negatively charged group and nonpolar tail. So through hydrophobic interactions, it's going to bind to the proteins. So let's do a quick review. If we load samples at the top and the proteins move down from top to bottom, what are the different electrons? Where is plus? Where is minus? Pause for a moment, see if you can answer that. So moving from negative towards positive electrode, right? And then where are the large and small proteins going to be? Large are going to travel shorter distance, whereas small are going to travel much longer, right? Because it's easier for them to get through. This is going to be an important distinction when we talk about purification method with how we separate by size. All right, so you can also put the two methods together into two-dimensional gel electrophoresis. First dimension separate by pi, second by size. And when you see a gel like that, what can you conclude about a protein? So if we look at this protein right here, right, we can say, oh, it probably has pi around 6.5, and its size is a little under 30 kilodaltons, maybe 27. It's okay to guess, right? And it has other proteins around that will vary by pi and by size. All right, so real proteins or real 2D gels look a little more complicated when you take unpurified sample. But what's important is that this allows you to kind of take a really complex sample that on just SDS-PAGE you would see, imagine all these bands vertically, you would see just a smear of dark bands, right? It allows you to separate this really complex mixture by two properties, and then you can compare samples. So for example, you can compare normal tissue with a tumor tissue and see which proteins change either expression level or their phosphorylation, because their mobility will be affected in this analytical method. So next we're going to think about detection of proteins, because what you see here is a gel that's been stained, because normally most proteins are not colorful. They're not visible. Think about egg white, right? It might have a little yellow tinge there, but it's mostly clear before it's cooked. And so when we want to detect protein, we have choices. most non-destructive method is just to measure absorbance using spectrophotometry, because proteins, almost all proteins, absorb UV light at 280 nanometers a max. You can see there's kind of a peak, and that peak comes from amino acid side chains that have conjugated double bonds like tryptophan, tyrosine, histidine. And so recall that Beer-Lambert law allows you to correlate absorbance to concentration. So if you knew absorptivity or molar absorptivity for your protein, you could even quantify it. Now, a lot of the time, we don't know what absorptivity is, especially if it's a protein we're purifying for the first time. But then you can just kind of use rough estimate, or you can do relative amounts. So a lot of the time people use about 1 or 1.5 for the absorptivity if you want to get concentration in mg per ml. Okay. All right. So in addition to A280, you can also just stain proteins with a dye, and Commassie Blue is a dye that stains proteins really well. So when you start with clear gel, if you stain it, you'll see blue bands, right? And this Commassie Blue dye can also be used in solution. So if you this is a 96 well plate, so when you have a lot of samples, you can often use that to read absorbance of many samples all at once. And you can see the samples that have blue color are the ones that contain a lot of protein, a little bit less blue, less protein, no blue, no protein, right? And if you have a set of standards, proteins with known concentration, like usually BSA is used for the standard, then you can compare your protein to a standard, generate standard curve, and quantify your protein in the sample. In addition, we can detect proteins using specific antibodies if you have an antibody for your protein of interest. And the method that we can use that goes with SDS-PAGE is Western blot. It just requires another step where after you separate proteins on a gel, you transfer them onto a membrane that you can then probe with an antibody. And that is shown here. So we're now talking about immunochemistry using very specific interactions with antibodies. Recall how precisely antibody fits with complementary surface to the antigen, right? So in Western blot, you start out with your protein that was separated by size, immobilized on the membrane, and then usually there are other proteins around, and the first step is usually block non-specific binding. Often people use BSA or just milk proteins. They're easy and cheap to get, and they will just kind of cover sticky spots. So that after that blocking step, you incubate your membrane with your specific primary antibody, and because of the blocking, it should not bind just to sticky spots randomly. It will bind to its specific target. You may have to incubate for a little while to give it a chance, right? But then you still can't see anything because antibodies are also colorless. And so there's usually a second step where you add secondary antibody, and that one is specific for the constant domain of the previous antibody. So if your specific antibody for your protein of interest was made in rabbit, all rabbit antibodies will have the same constant domain, and so you need anti-rabbit or rabbit-specific antibody that's made in another animal, like chicken, goat, whatever, right? But what's special about the secondary antibody is it's linked covalently to an enzyme, usually horseradish peroxidase, again, kind of a cheap, easy to get enzyme. And horseradish peroxidase catalyzes reaction with a number of substrate, including some that produce colorful products. So it can be yellow, it can be blue, right? And so what we can detect is actually product of enzyme catalyzed reaction. So we have to add substrate and wait to see the development of the color. Also, modern techniques allow you to do chemiluminescence or fluorescence, but you need a special detector for that, to read that. Right? So notice in this situation, for each protein that we kind of immobilized on a membrane, we're going to have millions of colorful molecules produced, the molecules we can detect. So the enzyme catalyzes millions of reactions to amplify the signal. And when I say amplify the signal, one molecule down here will give you many molecules that you can detect. Right? So this is the source of sensitivity of both Western blot and ELISA, which works very similar, except it's not on the membrane. It's usually done in a 96 well plate. And we'll talk about ELISA in another lab. All right, so these are our analytical methods. We're now going to switch and focus mainly on ion exchange chromatography and gel filtration chromatography. So in gel filtration chromatography, we're going to have chromatography column into which we're going to pour our sample that contains multiple proteins in it. And we're going to let the liquid run through. There are different methods you can pump it through, or you can just let gravity drip. And our samples are going to come out the bottom, and we're going to collect fractions so we can separate them. So when we separate by size, right, different size proteins will come out at different times, so they will end up in different fractions. So these tubes here represent fractions collected. Now let's think about what's in this column that allows a separation by size. We're going to see that the column is packed with beads, and these beads are usually either agarose or acrylamide, similar to what you saw in SDS-PAGE. The matrix might be similar, or some other polymer that has channels inside these beads. So the name size exclusion is probably more appropriate, because large proteins will be too large to fit into these channels, and they'll travel around the beads and come out first. So large molecules do not enter the beads and travel around them. So they are excluded because they're too large. Small molecules will kind of travel through the beads, and so take longer paths to get out. And depending on sort of some intermediate molecules will go a little bit and kind of come around. So that's our separation by size. Large come out first, small come out last. And it's not that they have different speed, it's just that they travel different distance. Because small proteins, and by the way, nothing is unfolded here. The structure hopefully is not disrupted, right? So multimeric proteins will stay attached with multiple subunits together and travel as large proteins. But small proteins come out last. All right, so when we do this kind of experiment, we collect fractions, and guess what? We can monitor presence of proteins by measuring absorbance at 280, right? It's very non-invasive. Many chromatography machines are equipped with UV detectors, so you're kind of reading absorbance as stuff comes through. If you don't have that, you may have to measure by hand, just take samples. All right, so what this chromatogram tells me is fraction 25 contains a peak with high absorbance. I'm going to assume absorbance proportional to concentration, has a lot of protein, right? Then I have a little bit less, but there's still stuff around here. And then there's another big peak at 55, fraction 55. These are just fraction numbers. If each fraction is 1 milliliter, this would correspond to the volume, or we'll just go by fraction. right? And then down here, looks like everything came out. There's almost no protein there. We can take these fractions and run them on SDS-PAGE. And what do we expect, right? We expect early fractions to contain large proteins, later fractions to contain small proteins. So if you look at the simulated gel, it seems consistent, right? So fraction 70 here has low molecular weight proteins, fraction 40 has higher molecular weight. Down here, we have a big mix, and that's probably corresponding to fraction 20 through 30, right? That's probably all the stuff that got excluded from the beads. So we don't get much separation. It just all comes out pretty much all together. And my question for you is, why might you get really low molecular weight proteins? Why aren't these bands coming somewhere over here in the fraction 60 or so, right? Why are they coming in the excluded volume? Hopefully you paused and thought for a little bit, right? Hopefully you came up with the idea that these chains on SDS-PAGE gel, right, may have been attached or to larger chains, or they may form multimeric proteins, right? Proteins that have quaternary structure. All right, so the media or the material can be different. So dextran, acrylamide, or agarose, or some mixture of them. And different columns will have different optimal size that they can separate. Usually you just kind of try to adjust for what you want to separate. So for example, this Biogel P60 separates from 3,000 to 60,000 daltons, so 3 to 60 kilodaltons. And then there is a gel with different size pores or different amount of cross-link on polyacrylamide, 15,000 to 150, or there's even one with probably larger pores that separate 60 to 400 kilodaltons. And this sort of what usually the apparatus looks like. The columns are pretty long. There is some kind of machine that runs the liquid through, and you either collect fractions by hand or you have fraction collector. By the way, this often is done in a cold room because proteins tend to unfold when they're warmed up or get degraded because proteases are active. And so a lot of these experiments are done in a cold room. So it's basically like a refrigerator you can walk into, and it's not very pleasant to do that. All right, quick check. What do size exclusion chromatography and SDS-PAGE have in common? They both separate proteins by size, right? But what do they have different? So let's review that. SDS-PAGE, small travels fast, right? Size exclusion, small eludes last. Okay. Next, we're going to switch to ion exchange chromatography. And this is a picture of, again, a setup. Usually there's a computer that's running it. There is some kind of fluidic system that pumps the liquids, also detector that reads UV absorbance. This machine has the automated fraction collector, so it's just going to kind of have a place where proteins drip out of the column and they're collected into tubes, and the machine advances the tubes automatically. Okay, and the main principle here is opposites attract. Right, so the idea is you're going to have charged beads in your column. Positively charged proteins will bind negatively charged beads, and negatively charged protein will flow through. Right, and when we're thinking about it, the question becomes, well, what pH buffer should you use? What should the charge of the protein be at that pH? And I'm hoping you're thinking, oh, we need to know pI of the protein, right? And so once you know the pI, you can start thinking, well, what column do I want to choose for doing this experiment? And there's often not an easy solution. You can have to match up the pH of your buffer to what the protein likes, so it doesn't get unfolded in what column you have, right? So the logic here is something like if the pH is below the pi, then we have protein more protonated than its Zwitter ion that exists at pi, and a more protonated protein will be positively charged, so it binds negatively charged column. All right, so once you bind your protein, so imagine we collected the stuff that eluted, pink protein is gone. Now we have a protein we want and other proteins that are also charged, positively charged. They're all bound to the column. So how would we elute this? The protein of the column, right? And you should be thinking kind of two things here. We have ionic interaction. How can you disrupt ionic interaction, right? And from what we know about ionization of protein side chains, you should be thinking, well, I can change the charge on the protein, right, by changing pH, or I can compete out this ionic interaction by adding some more ions, right? So, for example, sodium chloride. And so typically, researchers will use salt because it's generally, fairly gentle. With changing pH, you have to worry about protein falling out of solution or losing activity because of denaturation. But for some proteins, they don't like to be in a salty solution, and then pH is really the only thing you can do. And there's different media, of course. So we have positively charged columns, examples DAE or Q, or negatively charged columns, like something that has carboxyl group or sulfate groups on it. And they have like different pH range where they work best. So with this, let's do a first practice question. If you have a mixture of three proteins, PI5, PI8, and PI9.5, you have negatively charged column, and you want to purify the blue protein, which has pi of 8. What pH buffer should you use for that? So this is where I want you to pause the recording, try to answer it before you move forward. All right, so along those lines, we should be thinking about which protein will bind from this mixture. So I'm hoping you picked something like buffer pH 7 might separate these, right? So let's say if we go with buffer pH of 7, so at least some proteins are below this pi and some are above. So which proteins will bind column, which will not, which flow through? If we elute with salt, which protein elutes first? Again, try to think through this on your own before you go further where I explain the answer. And you can be thinking back about the column we saw earlier, right? So the pink protein at pH 7 will be at pH above the pi, so deprotonated compared to net charge of 0. So pink protein will have net negative charge and flow right through, right? Whereas blue and green will have net positive charge and bind. If we elute with salt gradient, which protein will come off first? This is a little bit harder question, right? And you can think about the closer the pH to the pi, the smaller the charge. The further away the pH is from pi, the larger the charge. Hopefully that makes sense in terms of protonated, getting deprotonated, right? So based on that, whoops, that didn't work well. All right, so the chromatogram from ion exchange would look a little bit different. And on this chromatogram, we are monitoring absorbance at 280, that's a blue line. But we also have salt gradient, which is my pink line. So you'll notice first when we're loading stuff on the column, the salt is low. And somewhere around fraction 30, maybe 32, we start increasing salt, and it goes from, say, 10 millimolar to 500 millimolar. or 0.5 molar, right? And as the salt increases, proteins come off, and I see 2 peaks here. There's probably one more. So hopefully what you can predict is what didn't bind came off before salt was added. This would be pink protein with pH5. And then the order of elution would be blue, then green, right? Because our buffer pH is 7, so pI of 8, the blue protein is 1 pH unit away from pI, so it's a little bit protonated, but not too much. But the green protein is 2.5 units, pH units away from pI, so it's very charged. And the larger the charge, the stronger the ionic bond. All right, so practice 2. Again, I recommend reading the question and then pause the recording and try to work it out on your own. This goes back to size exclusion chromatography, and I want you to think about familiar proteins. Hemoglobin, myoglobin, and IgG are in a mixed solution, and we want to separate them. So we choose size exclusion chromatography, and we want to understand. We get 3 peaks. Well, good, we separated them. Can you label which peak is which? The earlier peaks have lower fraction numbers. The peak that came off last will have larger fraction number. Okay, and also, could you sketch what you would see on SDS-PAGE if you took fraction 25, fraction 50, and fraction 70 and ran them on the gel? So see if you can sketch what that looks like or predict band sizes. All right, so here's the answer I would get. I expected fraction 25 to get 2 bands. I expect that my fraction 50 and 70 will have single band that's fairly low, right? And so here's how I would label these. So this is a quick check on, do you remember that IgG is composed of 4 protein chains, 2 heavy, 2 light, right? So it gives you 2 bands, 50 and 25 kilodaltons, while hemoglobin and myoglobin have very similar primary, secondary, tertiary structure, right, about 15 kilodalton. But then why did they separate in size exclusion, right? And why did myoglobin come out last? It's because hemoglobin is tetramer, right? So in size exclusion chromatography, it's actually running as 60 kilodalton protein, whereas myoglobin is just 1 subunit. While on SDS-PAGE, we are going to separate all the chains. I hope you enjoyed this practice. This lecture should provide enough background for you to try simulated protein purification, and we'll work more on that in lab.